Hi, this is Kristen Haymond, and we're going to do a quick overview of flow cytometry. What it is, how it works, and what are some of the uses of it, especially in immunology. Flow cytometry is a technology that enables single cells or particles to be charted and analyzed after they are diluted and suspended in a salt-based buffer. As the cells flow past ultraviolet lasers, two things are measured by a detector. The detector measures the light they displace, or, quote, scatter, end quote, and also it measures any fluorescence parameters. Visible light scatter is measured in two different directions, the forward direction, which basically measures the relative size of the cell, and at a 90-degree direction, or side scatter, which measures the internal anatomy or granularity of the cell. And those things are revealed by side scatter. Fluorescence refers to light emitted from chemical compounds called fluorescent markers, and it's used to detect cellular molecules such as things like proteins, nucleic acids, and other things like that that are expressed in the cell. They're designed to absorb light energy at one wavelength, and then they actually re-emit it at a longer wavelength. Samples of cells are prepared by transfection, which is the infusion of the cell with nucleic acids, or the use of fluorescent proteins, dyes, or fluorescently conjugated antibodies. What is a fluorescence activated cell sorter, or FACS? This is some information from a website called NanoCollect Biomedical. And so basically, fluorescence activated cell sorter is a subset of flow cytometry technique that uses specific antibodies labeled with a fluorescent molecule called fluorochrome. And FACS basically analyzes for data collection and sorting of biological samples from a nearly limitless number of parameters, and those parameters can be set by the operator. So depending what they're looking for, it can be adjusted. You can see here in this image that the flow cytometer machine is used to suspend cells or other particles in a liquid stream, same as before, and passes through a laser. And the laser interaction with the laser light is measured by electronic light detector apparatus as light scatter and fluorescence intensity. If a fluorescent marker is bound by a cellular component, the amount of fluorescence intensity will represent the amount of that particular component in that particular cell. And then also the cell sorting machine uses electromagnets to impose an electrical charge on each cell to sort them by electrical charge. Can you imagine what would happen if we did that with our laundry? Anyway, once sorted, the cells leave the chamber and enter separate containers. So what data can be represented on these scatter plots that are produced through flow cytometry? Well, we can learn about cell count and size. We can also learn about cell type. So that would be differentiation of normal cells. You've got your monocytes, your lymphocytes, granulocytes. You can see those are represented on section B there. You can find cancer cells, etc. You can also learn about the internal components, things like what are some of the intracellular proteins of granulation, RNA, etc. Also, we can learn about membrane receptors and other protein expression. Catherine McKinnon says that one way of flow cytometry analysis, the conventional way, consists of drawing a region around a population of cells, that's called gating, and then applying that region to other parameters within the experiment to further select and analyze and narrow the pool of whatever you're looking for. For example, helper T cells can first be defined by CD3 and CD4 expression, and then they can also be analyzed for activation by looking at that specific population for expression of a certain activation marker or perhaps a cytokine production. There are a lot of applications for flow cytometry in immunology, and again, some information from Ms. McKinnon. Immunophenotyping is one thing that flow cytometry can help with. We can find genetic defects. Antigen-specific responses. Little information on this one. Antigen-specific responses can be measured by stimulating cells with a specific antigen and then looking for cytokine production, proliferation, activation, memory, or antigen recognition. And then intracellular cytokine analysis is performed by treating cells with a protein transport inhibitor for 2 to 12 hours so that cytokines produced by the cells can accumulate for better detection. The cells can be stimulated with various antigens during this incubation, such as peptides from a vaccine to measure immune response. Following protein transport inhibitor treatment, cells are stained for viability markers and cell surface markers, and then fixed and permeabilized for intracellular staining with anti-cytokine antibodies. Here are my references and image credits. Thanks for listening.